Welcome to TIT VKT. As requested, today I have come with the topic cell biology, and today we will be discussing on it. So first of all, let's discuss on the types of cells. This is a prokaryotic cell. Well, this is a circular non-stunned DNA. This is mesosome. This is ribosome. This is called flagella, and we have membrane. This is capsid or the protein coat or slime layer, and then we have cell membrane or cell wall. So this is just a normal structure of a prokaryotic cell. Well, this is an animal cell. This one is a nucleus, and it is surrounded by rough endoplasmic reticulum. This is a nucleus. This is lysosome, also called the suicidal bag, and this is mitochondria. And then we have golgi apparatus or golgi body or golgi vesicles. Golgi apparatus, and then we have vacuoles. This is a normal structure of animal cell. This is a prokaryotic, and these two are eukaryotic cells. So then finally let's talk on this cell. This is nucleus. This large structure is vacuole. These are golgi bodies, golgi apparatus, and then this is plastid, which is present only in plant cell, and then this is mitochondria. This is cell wall. In animal cell only cell membrane is present but no cell wall, but in plant cell both cell wall and cell membrane are present. Two plant cells are connected through plasmodesmata. This is a plant cell. Well, we discussed about cells, but mainly in entrance exams only names are asked from this chapter in IOM, KU and other entrance examinations in Nepal, and there are matching questions from cell biology, especially in NEET exams of India. So let's start with the cell. Cell firstly discovered by Robert Hooke, and first living cell was seen by A.V. Leeuwenhoek, also called father of microscopy. Now cytology. Cytology means the study of cell. The term cytology was coined by Hartwig, and then the father of cytology is the one who discovered cell, means Robert Hooke. He discovered cell from phylum of clerk, and he has included in his book Micrographia. So these are the things you need to remember. The things will continue. Modern father of cytology, Swanson, and he is also the father of cell biology. And there is cytogenetics, termed by Muller. Muller, famous football player, you can remember like that. And then animal cell, longest animal cell, nerve cell, largest animal cell, ostrich egg. Plant cell, longest plant cell, ramifibers. Largest plant cell, acetabularria. Acetabularria is umbrella shaped. And then we have cell theory. Cell theory says omnis cellula is cellulae, which means all cells are made from pre-existing cells. But this is cell lineage theory, and to make cell theory we have to add this cell lineage theory with old cell theory given by Theodor Schwarzman and Skeleton. Now talking about cell wall. Now we are entering into the cell. Cell wall discovered by Robert Hooke, who called it diaphragm. He saw honeycomb-like structures while seeing the phylum of clerk. Cell wall, middle lamella is formed of calcium and magnesium pectate. Then primary cell wall, elastic due to deposition of hemicellulose. Secondary cell wall, inelastic due to lignin. Now talking about protoplast. Protoplast is cell minus cell wall. Other thing except cell wall. Formed by Einstein. And then cell membrane. Inside of cell wall in plant cell and outermost covering of animal cell is cell membrane, discovered by Schwarzman, one giving cell theory, termed as cell membrane by Nageli and Kramer, termed as plasma membrane by Pfeiffer and plasma lemma by Jeffries, termed as cytoplasmic membrane by Singer and Nicholson. So it's like a lecture only, because there are only names. The important names are this and this. You may or may not remember this, because only names are asked sometimes only. Now models of cell membrane. Unit membrane model given by Robertson. Only one son. Unit Robertson. And lipid model Overton. Lipids Overton. And then we have sandwich model Danieli and Epson. These are all to remember. Fluid mosaic model by Singer and Nicholson. Flute is played by Singer and Nicholson. If you Singer and Nicholson. Cell wall impermeable in cork shell and Casparian stripes due to deposition of suberin. Cell membrane. Then now change into mesosome in bacteria. Mesosome respiratory function in bacteria. Lomasomes in fungi. Lamellosome in blue-green algae, and sometimes microvilli in intestine of animals. Now about protoplasm. Protoplasm means cytoplasm plus nucleus. That means we have to remove cell membrane from protoplast. Now in case of protoplasm, the most important question is who termed protoplasm. It was by Purkinje. P. Easy also. And observed by Alfonso D. Corti, termed as sarcodes by Dujardin TD, and then Huxley described protoplasm as physical basis of life. Due to plasm, non-living component of protoplasm, and cytoplasm minus cytoplasmic structures gives cytoplasm minus cytoplasmic structure gives hyloplasm. Now we have completed on protoplasm. Now let's discuss on cell component. Largest cell component nucleus. Largest cell organelle plastid. Largest cell organelle animal means mitochondria. Smallest cell organelle ribosome, peroxisome, smallest cell component means microtubules, largest microbody glyoxysome, organelle of plant cell, only plastid, and glyoxysome. No membrane bound structure, comprises of microtubules and filaments. Centrosome, ribosome, cilia and flagella. Triple membrane bound structure, transosome. Now we'll be discussing on plastids. Plastid was discovered and termed by Hegel, Heckl. Plastid also called plastid types: leucoplast, chromoplast, and then chloroplast. Chromoplast, anthocyanin or anthoxanthin is also present in Hegel. This give color pigment to the plants, which are water soluble. Plant color, give water soluble plant color. Chloroplast. Now there are different shapes of chloroplast, starting with spiral. Spiral, we all know that spiro gyra. Griddle shaped eulothrix. Second last. Star, zygnema. Cup shaped chlamydomonas. Reticulate, oidocnema. CCSS. Second last eye. Star shaped zygnema, you have to remember this. CC, cup shaped chlamydomonas. Reticulate, oidocneum, you have to remember this. These are important. In plastid there are two sites: stroma, site of dark reaction, and grana, site of light reaction. Quantasomes unit combine to form thylakoid. Thylakoid are stacked to form grana. Thylakoids are named by Menke. Thylakoid, if not stacked, called chromatophores or lamellar chloroplast. Now we will be discussing on mitochondria. Mitochondria absent in sieve tubes, cancerous cell and mature mammalian RBC except camel and llama. Yellowish color of mitochondria is due to riboflavin rich in manganese. Spermatids have mitochondria without cristae. Cristae are finger-like projections in mitochondria. This is the membrane, and then we have like this structure, and there are some oxysomes in mitochondria. So starting with cristae. Cristae have elementary cristae, then elementary particles or oxysomes, or called Fernandez-Morgan particle or electron transport system. Mitochondria is powerhouse of cell, kitchen of cell, plastid. Powerhouse of cell, mitochondria. About 70% enzymes of cell found in mitochondria. Mitochondria together complete mitochondria of a cell are called chondriosome. This oxysome has 3 parts head, F1 particle, then has ATPase, tail F0 particle has proton channels of oxysomes. Important in mitochondria. And there is one name in mitochondria, Benda, who coined the term mitochondrion. Is important, coined or termed. And then absent in this is important, 70% enzyme important, and then cristae and oxysomes important in mitochondria. Now let's discuss on endoplasmic reticulum. Endoplasmic reticulum is termed by Porter and Kalam. It consists of 3 parts: cisternae, tubules, and vesicles. Cistern unbranched. Protein synthesis rich in ribosomes. Protein synthesis branched. Transportation of material inside a cell. Material inside cell. Vesicles are spherical and store synthetic materials. Here are isolated, fragmented form are isolated and fragmented to form microsomes. Then it is also called membrane factory of cell. Membrane factory. And myeloid bodies and Nucleus granules are mass of smooth endoplasmic reticulum and rough endoplasmic reticulum. Important membrane factory, myeloid bodies, Nucleus granules, cistern tubules, vesicles stormed by porter and column. Important in endoplasmic reticulum. Easy in Golgi bodies. Golgi bodies are stormed by Golgi and found in Purkinje cells of cerebral cortex of barn owl. Golgi bodies is called dictyosome in plants. There is one famous saying for Golgi bodies: bad middle man to dig. Bad middleman to dig. B. Baker's body. Dictyosome, middleman. And then D. Dalton complex. Idiosome and Golgi body. So bad middleman to dig. Cis or forming face, which is towards membrane. Present in outer and convex in shape. So it will be like this. Outer convex. So here it will be membrane, here it will be nucleus. This is cis and this is trans or maturing face towards nucleus, concave inner. It is called traffic police of cell. Acrosome or head of sperm and yolk derived from Golgi body. Form zymogen granules. So it's something, it goes on and is like a lecture. 3 parts: tubules, vesicles, and vacuoles. Or we can also write here cisternae. Tubules form complicated network between transphase and peripherals. Vesicles contain secretory substance and vacuole function as lysosome. Lysosome are derived from vacuoles of Golgi bodies. Now one smallest organelle, that is ribosome. Ribosome termed by cloud, discovered by Palade. Cloud termed as microsomes. Cloud termed ribosome as microsomes, and then it was termed by robot. It is protein factory of cell. Factory, or also called engine of cell. Engine of cell. Found, most abundant, found in all except RBC and sperm. Absent in RBC and sperm. There are 2 types of ribosome, 80S and 70S ribosome. 80S ribosome 60S plus 40S subunits, and then 50S plus 30S subunits. And there are 3 types of ribosome: rRNA, mRNA, and tRNA. Ribosomal RNA, messenger RNA, and transfer RNA. 80S and 70S are the types of rRNA. rRNA is the most abundant, about 80%, and there is mRNA, highest molecular weight highest, and least abundant, only about 5 to 10%. And then we have tRNA, smallest RNA, with covering 10 to 15%. Now on lysosome. Lysosome, also called suicidal bag of plant, discovered by D. Dube from rat liver cell. There are 3 types of primary lysosome, and then secondary, and then finally residual bodies. These are digesting. This lysosome will be also talking about spherosome. Spherosome, also called plant lysosome or oleosome, discovered by Perner and termed by Dangiard. Now we'll be discussing about microbodies, which are of 2 types, peroxisome and glyoxysome. Sometimes even lysosomes are also kept under microbodies. Peroxisome. Peroxisome discovered by Rodin. Rodin from kidney cell. And then Tolbert and Yamazaki discovered it from plant cell of germinating seed. Termed by Buffet and Berther. These are quite not important. And then on glyoxysome, largest microbody found only on plant cell. Beta oxidation in plants occur in glyoxysome. Beta oxidation occur in plant, but beta oxidation in animal occur in, yes, it's in peroxisome. Now there are some special terms like lomasomes. Yes, and we had discussed earlier lomasomes in fungi. Discovered by Moore and McAllister. Help in cell elongation. Transosome found in ovary follicle cells of bud. Helps in yolk formation or vitellogenesis. Microtubules discovered by Robertis and Franchi in nerve fiber as tubulin protein named or termed by Slaughterback. Basic structure of spindle apparatus, centrioles, chromosome factor. Now then microfilament protein filament made up of actin protein. Now on centriole membranous list found in animals except amoeba, animals and plants except red algae and gymnosperms. Centriole responsible for microtubules, organization, and spindle formation during cell division. Now on flagella. Vessel granules, all Phaloplast is kinetic center of flagella that lying in protoplasm. Lying in protoplasm. Prokaryotic flagella is made up of prokaryotic flagella made up of flagellin protein and eukaryotic flagella made up of microtubule. So in eukaryotes microtubule, in prokaryotes flagellin protein, Phaloplast is the kinetic center of flagella. Let's continue with vacuole tonoplasm inside of vacuum, which is non-living, bounded by living semi-permeable tonoplast. And then we have food vacuoles. In protozoans, food vacuole, food material association, associated gas vacuoles. Gas vacuoles helps in contractile vacuoles helps in osmoregulation in protozoans. And in prokaryotes, gaseous exchange takes place through gas vacuoles. And then we have sap vacuole. Higher plants, turgidity. Now finally we have nucleus. Nucleus was discovered by Robert Brown from orchid root. Discovered. Hammering proved nucleus as the director of cell by hammering emerging from grafting experiment on Aceta bullaria absent in sieve tube and mature mammalian RBC except camel and LLA llama. Nuclear envelope which covers or is called karyotheca, which was discovered by Hartwig. And there in the inside of nuclear envelope we have nucleoplasm, discovered by Fontana and named by Bowman. And then we have nucleolus, LUS, nucleolus. Sorry, this is nucleolus. Nucleolus was discovered by Fontana and named by Bowman. And nucleoplasm, no cyclosis. Cyclosis, that is movement of cytoplasm, does not occur in nucleoplasm. And then it is rich in histone protein. Nucleolus is also called ribosome factory in eukaryotes. Ribosome factory in eukaryotes, nucleolus. And then we have chromatin fibers or nuclear reticulum, discovered by Waldier. Who coined the term chromosome? Waldier. On the basis of stain, chromatin fiber are heterochromatin and euchromatin. Hetero thick, this thin, narrow, thin. Yeah. Don't take part in transcription. No active gene inhibit crossing over. Here crossing over common. This is active gene. Active crossing over. No active gene inhibit crossing over. Now we are finally discussing on chromosome. Chromosome important, discovered by Strasburger. So this Burger who discovered chromosome and then coined by as I told Waldier. Chromosome has two chromatids made from chromonimata, two chromatids. Secondary constriction containing chromosome with secondary constriction. Set, all set. Now there are shapes of chromosome: I, J, V, L, telo, acro, sub, meta. These are all living components, and the non-living components are same. Inclusions. Cell inclusions include lipid, fat, carbohydrate. Let's not discuss on that. Well, as I have discussed today, it is whole on lecture and whole on remembering names and their functions. So you have to do it yourself. There is no specific shortcuts, but you can make mnemonics yourself, because making mnemonic on self will be more remembered. Thank you. This much for today. Keep tuned for more videos.